of the urinary system as well, as you wish. So maybe I just would propose to start with the embryological part, and then once we are finished with that, we can talk about the histology part as well, kind of repetition, if you agree. So first of all, I would like to ask you if you have any specific question regarding the development of the urinary system, or any specific topic suggestion to go through once again. It may mean two very different things. The optimist scenario that you are so well understood everything that you don't have anything to ask because you already know everything. Should be, but I don't know. Yes.
Process on slide 6. Can you explain that?
Slide 6, the pronephros development. Well, that's not a big deal. You know, 3 kidney systems develop during intrauterine life in craniocaudal order. Here in this drawing, all systems are visible, but in reality, all these three systems never coexist. The first one is the pronephros, what you are asking about, which is never functioning. So it develops partially, but never will function. This happens during weeks 3 and 4. And then the mesonephros, which will function for a short period of time, and it's collecting duct, the mesonephric ducts, it's a paired one, will persist in males, but will disappear mainly in females. And eventually it will, this mesonephros will disappear. And the permanent kidney is the metanephros, which starts to develop fast. So the pronephros is just in the cervical and maybe upper lumbar region, and they appear as a kind of cell accumulations which acquire a lumen, and a kind of collecting duct also appears, but actually these primitive excretory units will never function, and they will start to disappear by the time the next kidney system, the mesonephros, starts to appear. So this collecting duct may be called as pronephric duct very conveniently, but again it will disappear without any remnant. Yes. You are talking about the permanent kidney? No. No. The kidney is mesodermal. It is from the intermediate mesoderm of the intraembryonic mesoderm. So, you know, the intraembryonic mesoderm is divided into 3 major parts: paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. And this intermediate mesoderm is also called mononephrotome, by the way, because this name refers to the fact that at least most of the urogenital system is derived from this part of the mesoderm. So nothing to do with the endoderm. Endoderm contributes to the urinary bladder, to urethra, but not to the kidney. What do you mean all of them have the same origin? So this is what I should project here, I think. So this is the hindgut, and it ends in a dilated portion called cloaca. And there is an outgrowth of the cloaca, which is called allantois, which is present in the primitive umbilical cord. Now here, this grayish territory is the so-called urorectal septum, which starts to grow towards the cloaca, and this sequence from left to right shows as the urorectal septum is growing, and eventually we'll separate the cloaca into two parts. One part is the urogenital sinus, and the other part is the anorectal canal. And because the cloaca is a continuation of the hindgut, therefore the lining epithelium of the cloaca, just like the lining epithelium of the primitive gut, is endodermal. As a consequence, the lining epithelium of the urogenital sinus is also endodermal, right? Now, this urogenital sinus then gives rise to three parts. The upper part is the definitive urinary bladder, which is here. The pelvic part, which is here, in males gives rise to the prostatic and membranous part of the urethra. In female, it gives rise to the entire length of the urethra. In males, there is a third part, the Foley part, which will give rise to the penile urethra, part of the urethra which is in the corpus spongiosum in the penis. So because all these are coming from the urogenital sinus, which is coming from the cloaca, therefore all these parts of the urinary system has endodermal lining. Okay. But regarding the urinary bladder, there is a complication during development, because as you can see, at the beginning of the development, the two mesonephric ducts directly connected to the urinary bladder. So these are the excretory ducts of the mesonephric system. And there are outgrowths from each, which are the ureteric bud. Because the mesonephric duct has mesodermal origin, therefore the ureteric bud also has mesodermal origin, right? And because ureteric bud gives rise to the ureter and the collecting system of the permanent kidney, therefore all these parts of the urinary system has mesothermal lining epithelium. And the other thing that during development, as you can see, sooner or later the ureters will have direct openings into the urinary bladder instead of budding out of the mesonephric duct as it was originally. And the other thing that during development, these two mesonephric ducts are involved in development of the wall of the part of the wall of the urinary bladder, actually the trigone. The trigone is the triangular shaped territory over the surface of the urinary bladder whose corners are determined by the openings of the two ureters and the opening of the future urethra. So, because of this, for a limited period of time, the lining epithelium of the trigone has mesothermal origin because the mesonephric duct is mesothermal. Unlike the other parts of the urinary bladder, which are entirely from the cloaca, so that is endothermal. But later, even this trigone will be replaced by epithelium with endodermal origin. So eventually, by the time the urinary bladder is fully developed, its whole lining epithelium will have endodermal origin. So this trigone has mesodermal just temporarily. But interestingly, this is related, this developmental difference in the trigone relative to the other parts of the wall of the bladder is reflected gross anatomically and histologically as well. Gross anatomically, you know that this is the smooth part rather than the rest which is folded. And histologically, the trigone never has muscularis mucosae, but outside the trigone, in certain cases, you might find traces of muscularis mucosae, which is said to be related to the folded nature of the territory outside of the trigone. So here is a good example which illustrates how embryology is interrelated with gross anatomical appearance and histological features. So that's why we very like to ask this, because it's a nice example for this interrelationship, and we can measure how the student have a complex knowledge or not. Yes?
Let's say for the mesonephric ducts, there is no evidence of female in the trigone. Or is there any other structures that remain like the names?
You mean, is there any structure which is a derivative of the mesonephric duct? Yes. In the male genital system, there are certain ducts which are remnants of the mesonephric duct. We will learn this soon. Yeah. Anything else? At least one form of congenital polycystic is a advantage. That's true. It's here. This slide is about this. So, cysts may appear due to different reasons in kidneys. But one possible reason which may result in polycystic kidney, like these ones here, is due to the fact that during development in general, in many cases, and also during development of kidneys, certain special cilia on top of cells are involved in the development. This is called primary cilium, and is different from the kinocilium, the cilium which is found on top of lining epithelial cells of the trachea, for example, which can move. These other kind of cilia, called primary cilia, are different regarding its internal structure and also different in function. And also, one primary cilium is present almost on top of all cells. And it has been shown that these so-called primary cilia are involved in development of certain organs, like the kidney. And it is now known that if genes which encode proteins inside these primary cilia are mutated, so the proteins which are expressed will be mutant. Therefore, obviously, this primary cilium will not be able to function properly. Such an illness, which is related to this kind of illness of the cilium, of the primary cilium, these illnesses together are called ciliopathies. So, one form of polycystic kidney disease is a ciliopathy, because it is related to mutant genes which express proteins for the primary cilium. So that's the background, and here this is an illustration. In the middle you can see a normal kidney from an infant, from a newborn. On the two sides you can see kidneys which are polycystic, which are visibly considerably bigger. So I think you don't need to know more than this about this topic. And because this is a genetic disease, it may be inherited, of course. So that's it. For example, in the kidney, in the collecting duct, there are at least two types of lining epithelial cells. We don't teach this, actually, but I'm just telling because it is related to the primary cilium, because one of these types of lining epithelial cells has one primary cilium, and it is believed that this primary cilium can act as a mechanoreceptor and can sense the flow of urine inside the collecting duct. So, in certain other cases, it has mechanosensory function. And it is believed that this mechanosensory function may be related to certain deterministic pathways which need to work during proper development. So we don't know exactly, but maybe this mechanosensory function is the key why damaged primary cilia give rise to or related to polycystic kidney disease. Because in broader context, here you can see that the uretary bud is growing into the mesonephric blastema. It's an unsegmented part of the intermediate mesoderm. And in the meantime, the uretary bud is branching, like here you can see. And generally speaking, the uretary bud gives rise to the collecting system. So all these parts of the permanent kidney, ureter, renal pelvis, major and minor calyces, papillary duct, collecting duct, maybe connecting tubule as well. The rest, which is basically the excretory system, the nephron, is derived from the metanephric mesoderm, which is the caudal part of the intermediate mesoderm, which remains unsegmented, unlike the cranial part, which becomes segmented, and these segments are called nephrotomes. But such nephrotomes do not appear more caudally, because more caudally the intermediate mesoderm remains unsegmented and will be called as metanephric mesoderm. And this metanephric mesoderm, which is blue, gives rise to the excretory system, so basically the nephron, the tubular system of the nephron. But something very important needs to happen here, because the end of the uretary bud needs to interact with the metanephric mesoderm, the blue thing. During this interaction, such caps appear around the branches of the ureteric bud. Actually, these are metanephric tissue caps, okay? And during this interaction, the metanephric tissue cap gives rise to a S-shaped tubule, which is the primitive form of the tubular system of the nephron. And the one end of this S-shaped tubule needs to be connected with the branching ureteric bud. So actually the nephron, the excretory unit, needs to establish communication with the collecting system. So the ureteric bud needs to open freely into the developing tubular system of the nephron. Without this opening, which is here, continuation of the tubular system, without that opening, obviously if excretory system starts to function, but cannot, the excretum cannot be conducted away, it may give rise to cysts, enlarged cavities filled with excretory product. And this is highly important to understand here, because here the epithelium of the ureteric bud interacts with the mesenchyme of the metanephric mesoderm. And this interaction is in both ways, so that's why the double arrow here. And this is a so-called epithelial mesenchymal interaction, because the ureteric bud is already lined by an epithelioid tissue, so it's an epithelium. But the surrounding metanephric mesoderm is a mesenchyme, not an epithelial tissue. And there are two major differences, at least, between an epithelial tissue and a mesenchymal tissue. One is that in an epithelium, cells are interconnected by cell adhesion molecules. So they are all together. While mesenchymal cells are not interconnected. This is one difference. And the other major difference, that if we have an epithelium, it has a basement membrane around it. While during mesenchymal cells, there is no basement membrane. So when we are talking about here a kind of epithelioid transformation, how the mesenchyme of the metanephric mesoderm is transformed into epithelium, epithelioid transformation, it means essentially two things. One, that these individual mesenchymal cells will be joined together by cell adhesion molecules, intercell junctional complexes. And second, this is how the tubules start to appear, tubules of the nephron. Tubules is lined by epithelium. So cells will join together by cell adhesion molecules, and the other thing, that a special extracellular matrix appears these cells, which is this red circle, which is the basement membrane. That's a specialized piece of the extracellular matrix. So here's the expression pattern of cells, of these epithelioid cells, needs to change to be able to express those molecules which can later on give rise to the basement membrane. So we can say that this is a kind of epithelioid transformation, whereby the mesenchymal cells will become similar to epithelial cells eventually, because they will get joined together and will acquire a basement membrane around them. And this is how the tubule is formed, the tubules of the nephron. And this is a kind of interaction when an epithelioid tissue interacts with a mesenchymal tissue. And this happens here during development of the permanent kidney, but it also appears many times during intrauterine life, during development. For example, think about the formation of the paroxial mesoderm. Paroxial mesoderm will give rise to somites, which are kind of cell walls. These cells are interconnected, so they resemble epithelium in this respect. But later on, these somites, the cells of somites, will disintegrate, will give rise to sclerotome, for example, which are individual cells and will start to migrate and invade the territory around the neurotube. So in this case, the somite as an epithelioid tissue will become mesenchymal. But later, these sclerotome cells will start to form, for example, the vertebrae, as we learned. That's again something which is a structure which comprises cells which are interacting. So mesenchymal nature of these cells are more or less lost again. Another example, during gastrulation, primitive steak and primitive pit are formed, and the epithelioid cell mass of the epiblast will disintegrate, at least in territory where these cells start to migrate through the primitive steak and primitive pit and migrate to different parts and reside somewhere else. So during this, obviously the epithelioid cells of the epiblast needs to get disintegrated become mesenchymal, and then after migration they become epithelial-like cells again, because they will get joined together again. So this happens many times during. So that's why I feel this is important to know about this. And this kind of interaction, when one developing tissue is affecting the growth and differentiation of another tissue, is called induction in embryology, induction. So we can say that the ureteric bud induces the metanephric mesoderm to become epithelate. All right, and hopefully you already understand what all this means and what is the significance. Okay, what else we should talk about? Is there any other embryology what you would like to discuss with me? Or we can jump to histology. Jump to histology? Okay. So what would you like to ask about the histology part? So essentially we had just 2 seminars, I think, in this topic. One was dedicated to the kidney, with the 3 kidney sections, and the other seminar was dedicated to ureter, urinary bladder,